Good evening to you once again. The last time we met, we looked briefly at inhibition, enzyme. Before we should be looking at enzyme inhibition. The last time I mentioned that we should be looking at enzyme kinetics. So we looked briefly at enzyme kinetics, where we looked at the Michaelis-Menten equation, and then we also looked at the Lineweaver-Beck relationship, between concentration of substrate and the rate of enzymatic reaction. And we mentioned that the study of kinetics of enzymes is basically about the responses that enzymatic reactions have to changes in parameters that affect these enzymatic reactions. And these parameters which affect enzymatic reactions, we saw them as the concentration of enzyme or concentration of substrates. We saw factors such as pH, factors such as temperature. Of course, if you are doing some enzymatic study, sometimes you even will look at reaction time. So if you are studying the changes in the enzymatic reaction, depending on any of these factors, what you're doing is enzyme kinetics, or you are studying the kinetics of the activity of the enzyme. So today we'll continue to look at enzyme inhibition. Already, right from the first lecture, we looked at the term inhibitor, an inhibitor being component that either slows down a reaction or it terminates a reaction altogether. So today we'll look at that enzyme inhibitors much more, and then we'll look at the types of enzyme inhibition that we have. And under the types of enzyme inhibition, we'll look at reversible inhibition and irreversible inhibition. Under reversible inhibition, we'll have to break it down and look at what we refer to as competitive inhibition, then uncompetitive inhibition, then we look at mixed inhibition, and non-competitive inhibition. So these are the things that we'll be looking at today, the different types of inhibition that we have. So this is what today's lesson generally is about, to look at the types of enzyme inhibition that we come across in enzymatic activity. Right, so as I was saying, any molecule, any compound, any chemical entity, or any biomolecule that slows down or prevents the enzymatic reaction from proceeding, such a molecule, such a compound or biomolecule is an inhibitor of the enzyme that is catalyzing that specific reaction. So an inhibitor clearly acts on the enzyme. So sometimes some students make some error thinking that the inhibitor is acting on a substrate. No, the inhibitor acts on the enzyme. So the inhibitor either slows down the activity of the enzyme or it stops the activity of the enzyme. And by that, it will slow down the enzymatic reaction, the rate of the enzymatic reaction, or it will completely terminate the reaction from proceeding. And of course, such a knowledge of enzyme inhibitors is applied in the design of some pharmaceutical agents. So knowing that some enzyme is responsible for catalyzing a specific biomolecular reaction will, give you the idea that if you use a particular chemical or biomolecule, a compound, that can act as an inhibitor on that enzyme, you'll be able to control whatever activity that you want to control in the body with the drug. So we'll look at some specific examples where inhibitors are used as pharmaceutical agents, apart from the example which you see here with paracetamol and aspirin, which are painkillers. We refer to them as pain relievers, inhibiting the enzyme which catalyzes the synthesis of prostaglandins. And I think even in first year, you might have, when we talked about the fatty acids or lipids, you saw prostaglandins as some derivatives of fatty acids. So you have some fatty acids attached to some five-sided ring that will give you the structure of prostaglandins. And prostaglandins are responsible for the response of the body to pain. So they serve as some, they are recognized by the receptors of cells that are responsible for the feeling of pain in our bodies. So that if paracetamol and aspirin go to inhibit the enzymes that catalyze the synthesis of these prostaglandins, then of course you'll be relieved of pain. So that is an example of the use of inhibitors as a pharmaceutical agent. Then of course, if you want to study enzymes and have some valuable information about them, some inhibitors can actually be used in some research activities to study the activity of the enzyme. So you introduce, again, this is some form of enzyme kinetics. So inhibitors are introduced in a reaction mixture where an enzyme is present, and such an inhibitor, its activity is seen depending on the amount which is added in the reaction mixture. And knowing also the chemical structure of an inhibitor and knowing the properties of that and the chemical component which is serving as an inhibitor will give us an information about the type of reactions that occur in the active center of these enzymes. So for research purposes, yes, inhibitors are also used to study the mechanism of action of enzymes. Now, let's take the types of inhibition, enzyme inhibition that we have, one after the other. The first is referred to as competitive inhibition. And by competitive inhibition, what we mean is that you have here this diagram depicting what competitive inhibition is. So this is the enzyme that you see, the enzyme that is modeled here, the enzyme. And the active center is shown here. So this is the active center. This is where a substrate can bind with the enzyme for a reaction to be catalyzed by that enzyme on that substrate. So you see that in a normal situation, you have a substrate which is bound to the enzyme to give you an enzyme substrate complex. And then once you have an enzyme substrate complex, the reaction then proceeds at the speed at which it proceeds when an enzyme is introduced in that reaction. So you see that depicted here. as enzyme, and enzyme is depicted here as E, and then the substrate is S. So enzyme and a substrate, have an interaction, and they form an enzyme substrate complex, which you see here, the enzyme substrate complex. And then the reaction proceeds where you have a product out of that reaction, and then that product, when it is associated from the enzyme, you are left also with the free enzyme to catalyze another reaction, or to bind to another molecule of a substrate for the reaction to continue. But in a situation where you have an inhibitor which is present, what you realize is that the inhibitor looks very much like the substrate. So the inhibitor is some structural analog. It's a structural analog of the substrate. So the inhibitor can also bind to that active site. And if that inhibitor binds to the active site, what you have now will be an enzyme inhibitor complex. And once it is an enzyme inhibitor complex, the reaction does not proceed to give you product. And that is what you see here, where you have the enzyme E binding to I inhibitor and giving you an enzyme inhibitor complex. And thus, the reaction is terminated. The reaction does not continue. Now, you see that the substrate and the inhibitor are competing for the active site of the enzyme, the active site or the active center of the enzyme. So that is where the term competitive comes from, competitive inhibitor. Competitive inhibitor because the inhibitor is competing with the substrate for the active site of the enzyme. So that once the inhibitor binds to that active site, the substrate is not able to bind, and so the reaction is terminated. Or sometimes the reaction, you say, is slowed down. Because of course, with a competitive inhibition, if you continue to add more substrate into the reaction mixture, what you do is that you increase the probability of the enzyme binding to the substrate than to the inhibitor. So let me say that again. You remember that we separated the types of inhibition into reversible and irreversible inhibition. So we are looking at one of the reversible inhibitions, which is competitive inhibition. And I'm saying that if after you have an inhibitor bound to the active site and you continue to introduce more substrate in the reaction mixture, what you are doing is you are displacing the inhibitor which is bound to the active site already. Because, of course, that binding is reversible. And the more you add more substrate, you increase the probability of the enzyme binding to the substrate. So you can actually undo the activity of a competitive inhibitor just by introducing more substrate into the reaction mixture to go and displace the inhibitor. So the reaction can then continue, even all the way to the maximum rate of the reaction. All right. Now we see that this is a typical relationship that you see on the Lineweaver-Burke, relationship for competitive inhibition, where you see what happens is that when you increase the concentration of the inhibitor, the maximum rate of the reaction. So this, which represents the reciprocal of the maximum rate of reaction, you realize that it is a point of convergence for all these curves, even when you are increasing the concentration of the inhibitor. You see that these points on the vertical axis, which is the reciprocal of the maximum rate of reaction, is a converging point. It's a converging point because in competitive inhibition, the maximum rate of reaction is not affected. It's not affected because I explained earlier that if you have inhibitor present in the reaction mixture, have a competitive inhibitor present in the reaction mixture, and you continue to add more substrate in that reaction mixture, the substrate goes to displace the inhibitor and binds to the active site for the reaction to continue. So what it means is, if you continue to add more substrate, you can attain the maximum rate of the reaction. So that is why you don't have the maximum rate of the reaction being affected by a competitive inhibitor. You can still reach the maximum rate of reaction just by adding more substrate into that mixture. So you can say the maximum rate of reaction in a competitive inhibition is constant. The maximum rate of reaction in a competitive inhibition is constant, does not change. How about the apparent Michaelis-Menten constant? That is, you remember that the Michaelis-Menten constant is the concentration of the substrate at which the half of the maximum rate of reaction is attained. The concentration of the substrate at which half of the maximum rate of reaction is attained is what we saw earlier as the Michaelis-Menten constant. Now, with competitive inhibition, you see that even though you have the maximum rate of reaction being constant, what happens is you have to increase the concentration of the substrate in order to attain that maximum rate of reaction, which also means you have to increase the concentration of the substrate to reach half of the maximum rate of reaction. So what happens is that for the maximum rate of reaction is constant, but for the concentration of the substrate, you see it varying. You see that the concentration of the substrate will have to be increased in order to attain that maximum, the half of the maximum rate of the reaction for a competitive inhibitor. Now, how about what we refer to as uncompetitive inhibition? Uncompetitive inhibition. So here you see that, again, you look at the normal process of an enzyme that binds to a substrate S to give you an enzyme substrate complex, and then gives you a product that, when it dissociates from the enzyme, you have the free enzyme that can continue to catalyze the reaction. But then, after you have an enzyme substrate complex formed, you can have an inhibitor binding to that enzyme substrate complex. This time around, not to the active site, because of course the substrate is already bound to the active site of the enzyme, but the inhibitor binds to another site on the enzyme, generally referred to as the allosteric site of the enzyme. So you see it depicted well here for, more pictorially for you to see that this is an enzyme, and the enzyme has the active site. The active site is there, and it is having another site, the allosteric site, separate from it. And you see that the substrate binds to the active site of the enzyme, giving you the enzyme substrate complex. And then the inhibitor comes to bind to that enzyme substrate complex. So it will now give you enzyme substrate inhibitor complex. And this brings the reaction to termination. So the reaction is, the enzymatic reaction is terminated by the inhibitor binding to the enzyme substrate complex. So in this case, we refer to this as uncompetitive inhibition. Now, again, in an uncompetitive inhibition, when you are having a concentration of an inhibitor increasing in that reaction mixture, remember that the inhibitor is binding not to the active site, it is binding to an allosteric site on the enzyme. So it does not matter how much substrate you are adding the more into the reaction mixture. Even when you are adding the more substrate into the reaction mixture, the substrate will not go to displace the inhibitor because the inhibitor has its own site that it is binding to. So what it means is the maximum rate of the reaction will be lowered. It will be decreased because of the presence of the inhibitor. Because the inhibitor will easily bind to that allosteric site, which is free for it. The substrate does not bind to that place. And once you have inhibitor which is binding to an allosteric site on an enzyme, even though you have more substrate in it, still that effect of the inhibitor is there, and thus the maximum rate of the reaction is reduced. And if the maximum rate of the reaction is reduced, then it means the amount of the substrate that you add in that reaction mixture in order to attain that decreased maximum rate of reaction will also be decreased, meaning that the amount of substrate that you need to add in order to attain half of the maximum of the rate of reaction, which is the apparent Michaelis-Menten constant, will also be decreased. So this is really is logical. Really, it's not anything that complex. If you think about it, you have an inhibitor binding to an allosteric site. It means that it is going to inhibit the enzyme, even if you introduce more substrate into the enzyme reaction mixture. And so the maximum rate of the reaction will be lowered, it will be reduced, it will be decreased. And if it is decreased, you will need less amount of substrate to reach that maximum rate of reaction. And that also means you need less of the amount of substrate to reach half of the maximum rate of that reaction. And so you see that the apparent Michaelis-Menten constant is said to decrease. It's said to decrease, or it is said to be lowered, the same way as you have the maximum rate of reaction also decreased or lowered. You see this being depicted on, again, the Lineweaver-Beck relationship. Here you see that as you have the concentration of the inhibitor increasing, you don't see any convergence anywhere because the maximum rate of reaction, will keep changing, will keep decreasing as you add more substrates, you add more inhibitor into that reaction mixture. And remember that the value which is along that vertical axis is a reciprocal. And so you see that value of the maximum rate of the reaction actually getting decreased if you take that, if you take the reciprocal of that value. Then, of course, you also see that this is being depicted mathematically with some factors that are affecting the Michaelis, the apparent Michaelis methane constant, and at the same time affecting the concentration of the substrate. So you see the factor which is affecting the concentration of the substrate being depicted as alpha prime. We'll come to that when we come to the summary of this whole type of inhibition, which we are referring to as reversible inhibition. Right, now, we also have what we refer to as the mixed inhibition. And with mixed inhibition, really, what it is just uncompetitive inhibition, which is taking two different paths and can be occurring at the same time. And then we refer to that as mixed inhibition. So the uncompetitive inhibition, which we looked at earlier, took this path where you saw the substrate binding to the active site of the enzyme, first of all, to give you the enzyme substrate complex before the inhibitor now binds to the enzyme substrate complex to now give you the enzyme substrate inhibitor complex, causing the termination of the reaction. But there can be a second part where you have not the substrate binding to the active site first, but you have the inhibitor, which binds to that allosteric site first to give you an enzyme inhibitor complex. And once the inhibitor binds to that enzyme to give you the enzyme inhibitor complex, of course, the enzyme is already being inhibited. So even when a substrate comes to bind to that enzyme inhibitor complex, the reaction does not proceed. It just gives you that same complex, which is enzyme substrate inhibitor complex. So these are two different paths which are occurring at the same time. That is why it's being referred to as mixed inhibition. So you can say that mixed inhibition is uncompetitive inhibition, which is occurring with two different paths at the same time. And the first part is that which the enzyme binds with the substrate first before the inhibitor. The second part is where you have the inhibitor binding to the enzyme first before the substrate binds to the enzyme inhibitor complex. Both of these paths result into the same complex, which is the enzyme substrate inhibitor complex, resulting in the termination of the reaction. Now you see again here, because you are having uncompetitive inhibition that is occurring in two different paths, which we actually saw earlier. But you see that enzyme binding to an inhibitor before the substrate comes in, even though it's not binding to the active center, but the active center is still, open for the substrate to add to it. You have a mixed behavior in that reaction mixture when you are doing this experimentally and having a plot of the reciprocal, that Lineweaver-Beck plot where you have the reciprocal of the rate of reaction against the reciprocal of the concentration of the substrate. You notice that there is some convergence along the line, but that convergence is not on the vertical axis. And even that convergence, you see that it is actually lowered down from where you have the intersection on the vertical axis. As you increase the concentration of an inhibitor, that, of course, does not go to displace the substrate. It does not go even if you add more substrate into the reaction mixture, it does not go to displace the inhibitor, which is bound, as we saw earlier. And so, again, you have the maximum rate of the reaction decreasing. So even if there is a convergence along the path, that maximum rate of reaction is lowered, it is decreased. And that also means that the amount of substrate which is needed to reach half of the maximum is also decreased. So you see such a typical characteristics of the Lineweaver-Berg relationship with mixed inhibition, where you are having uncompetitive inhibition occurring by two different paths, as we saw earlier. There is that, there is that special situation where you have what is being referred to as non-competitive inhibition, which I refer to that you have that convergence somewhere, but it is lowered than where you have the intersections on the vertical axis. And the non-competitive inhibition is that for which, as I said, you saw that the changes that are occurring with the apparent Michaelis-Menten constant and the concentration of the substrate, the factors that affect that being referred to as alpha and alpha prime. If it so happens that in a mixed inhibition, you have these two factors being equal to 1, that is where you'll be having such a convergence. In that particular case, that inhibition is referred to as non-competitive inhibition. Not uncompetitive, but it's being referred to as non-competitive inhibition. Just for this particular case, where you are having that convergence there, which means you are having the effect on the Michaelis-Menten constant. That is the concentration of the substrate, which is needed to reach that reduced maximum rate of reaction. So that constant there is not the original Michaelis-Menten constant. That's why it's being referred to as apparent Michaelis-Menten constant, the concentration which is needed to reach half of the reduced maximum rate of reaction, which is caused by the inhibitor which is introduced in the reaction mixture. And the factor by which the concentration of the substrate is reduced in that reaction mixture in order to reach the maximum rate. You have the concentration of the substrate being reduced because, of course, the maximum rate of reaction is also reduced. You have that factor being referred to as alpha prime. So in summary of all of these reversible inhibition, you see it. So if you have in a normal reaction where you don't have any inhibitor present, so here you have inhibitor type. So in inhibitor, none, no inhibitor. The maximum rate of reaction being represented as Vmax, and the Michaelis-Menten constant is being represented by Km. In competitive inhibition, we said the maximum rate of the reaction is not affected, but the concentration of the substrate which is needed to reach half of the maximum rate of the reaction is reduced because you have the concentration of the substrate which is needed to reach half of the maximum of the maximum rate of the reaction is increased because you have an inhibitor present. So you have to introduce more substrate. And so you have that Michaelis-Menten constant, the concentration of the substrate needed to reach half of the maximum being increased by a factor alpha. And then you see for uncompetitive inhibition, we saw that the maximum rate of reaction is reduced. And it's being depicted here that it is reduced by a factor which is referred to as alpha prime. Similarly, the Michaelis-Menten constant, the concentration which is needed to reach half of the maximum, is also reduced by the same factor alpha prime. And for mixed inhibition, which is uncompetitive inhibition occurring by two different paths, you have the maximum rate of reaction also being reduced by a factor, which is being referred to as alpha prime. And because you are having two paths by which that uncompetitive inhibition is occurring, you are having this Michaelis-Menten constant also being reduced. But this time around, there are both factors are acting. You have the alpha and the alpha prime that is acting on that Michaelis-Menten constant at the same time, mathematically. All right, so that is a summary of the reversible inhibition. So clearly, reversible inhibition, as we have seen, we said the effect of the inhibitor can be reversed. It can be undone just by the name of the term. Reversible tells you that it is the interaction which is occurring between the enzyme and the inhibitor is not a permanent interaction. So usually what will happen is that the kind of bonding that exists between the inhibitor and the enzyme, in the case of these types of reversible inhibition, will be bonds like some weak hydrogen bonds, some van der Waals interaction, some weak electrostatic interactions. It can be some hydrophobic interactions. Any of these weak forces which can exist between molecules is what accounts for that reversible inhibition, so that it can easily be undone, can easily be reversed. Now we move on to irreversible inhibition. Irreversible inhibition. So to compare the reversible and the irreversible inhibition, you have here, this is an inhibitor, a reversible inhibitor, depicted in the orange color, and you see the arrow is bidirectional, meaning that the binding of the inhibitor to the enzyme, which is being depicted here as a protein target, is reversible. So it can bind, get on, and then get unbound, rebind. So you see that binding and unbinding and rebinding, all of that occurring in some equilibrium nature. So that is for what we saw earlier for reversible inhibition. But for irreversible inhibitors, irreversible inhibition, you have an inhibitor which binds so strongly with the enzyme that bond that is formed between the inhibitor and the enzyme is irreversible. It cannot be undone. So usually you have such bonding being some covalent bond which is formed. So it means that it forms a complex. It is forming almost like a molecule of its own. Say two molecules bound together covalently is turning into a different molecule altogether with some covalent bond between them. Or even if it is not a covalent bond, it is a stable, non-covalent, interaction between the inhibitor and the enzyme. So you can have very strong bonds, apart from the fact that yes, it can be some, if not ionic, Even if it is electrostatic, it must be a strong electrostatic force. If it is hydrogen bond, it might be some very strong hydrogen bond, and some other interactions in addition to such an electrostatic interaction that makes the binding of that protein target or the enzyme with the inhibitor so strong that it cannot be undone. All right, maybe it could be some ionic bonds, let's say, also, and then you have such very strong bonding, such very strong interaction between the inhibitor and the enzyme. All right, so a specific example of such strong binding between an enzyme and an inhibitor is what is depicted here between the enzyme chymotrypsin. Remember, we saw, I think in our last lecture, we saw the action of chymotrypsin, where chymotrypsin specifically breaks the bond between an amino acid, the carboxylic functional, so the peptide bond that exists between the carboxylic functional group of an amino acid and phenylalanine. So it could be an aromatic amino acid. So there is that specificity with that bond, which is between an amino acid, any amino acid, with an aromatic amino acid. That is that specificity we saw when we were looking at specificity of enzyme action. And we saw that with chymotrypsin. In our last lecture, there was I gave you a diagram where there was the activity of chymotrypsin step by step in breaking that bond in a polypeptide where phenylalanine was present. And you also saw that in that diagram, in the active center of chymotrypsin, there was the amino acid serine, and serine was at the 195th position in the sequence of amino acids in that polypeptide of chymotrypsin. So you can go back and refer to that, and you see serine 195 in the active center. Then you also remember that serine is having the side chain CH2OH. So you see the serine is being represented by the side chain CH2OH, and then the rest of the enzyme, the alpha carbon with the carboxylic acid group and the amine group, being referred to as ENZ, which is bound to CH2OH, which is the side chain of the serine molecule in that active center of chymotrypsin. Then we have the compound diisopropyl fluorophosphate, DIFP for short, DIFP, which acts as an inhibitor to the enzyme chymotrypsin. And this time around, DIFP is an irreversible inhibitor. An irreversible inhibitor because, so if this is DIFP, this is the fluorophosphate group, and you see that there are two isopropyl. So this is an isopropyl, this is another isopropyl. So you have diisopropyl, the fluorophosphate bound to them. And you see there is that displacement of the fluoride ion with a release of hydrogen ion. Then in that position where you see the fluoride, you now see that there is that conjugation with the enzyme chymotrypsin at the active center on that amino acid residues, the residue, the serine. So you see CH2, and then there is that bonding between the enzyme, covalent bond between the enzyme and DIFP. And that gives you a very strong association, very strong covalent association that cannot be broken. And so that kind of inhibition is irreversible. You know, DIFP is, apart from the fact that we are looking at it as an irreversible inhibitor to this specific enzyme, chymotrypsin. If you go to check, let's say, the label of many insecticides, you may see that you see some diisopropylphosphate. If the insecticide is an organophosphorus insecticide, so there are many of these organophosphorus insecticides where you have maybe some phosphate group, you know, or maybe even some fluorophosphate, for instance, like what you see in diisopropylphosphate, which acts as some nerve agents. So yeah, yes, they will act as nerve agents. I say nerve agents because they act on the central nervous system of the organism which they are acting. If it's because it's an insecticide, of course, it's supposed to act on an insect. So what happens is these substances actually go to inhibit the activity of acetylcholine esterase, which we also saw that acetylcholine esterase as a neurotransmitter is responsible for the contraction and relaxation of muscles. In that instance, what acetylcholine esterase does, we saw in detail. I mentioned the activity of acetylcholine and the action of acetylcholine esterase in hydrolyzing acetylcholine away from the muscle so that the muscle relaxes for a new signal to come before contraction happens again. So if you have, you know, such fluorophosphate, or you have an organophosphorus pesticide that goes to inhibit the activity of acetylcholine esterase, the result is what we refer to as paralysis, because then there will not be that signal for the muscles to be moving, the contraction and relaxation, because such inhibition of that organophosphorus compound on acetylcholine esterase is irreversible. So in effect, it causes paralysis on that organism. And of course, it's not that it cannot cause even such a paralysis in human beings. It can cause it in very high amount of concentrations. Such insecticides can cause paralysis in humans as well. And so sometimes it's really advisable not to be exposed to some of these insecticides. You know, the way we use insecticides indiscriminately. If you are chronically exposed to this, what happens is maybe now you may not be affected by it, but many of these organophosphorus compounds, they are lipophilic, meaning that they easily dissolve in lipids or fats and oils. So when they enter into the body, they can go and get deposited in the adipose tissue, for example, in fat cells, and will not be in the bloodstream. And so it will be as if they are not there and not having any effect. or the central nervous system. And later, in old age, if such a substance is accumulated in the fat cell, sometimes when there is some sort of a disease condition, especially when it results into weight loss, and weight loss will mean that there will be some breakdown of fat cells. And when there is that breakdown of the fat cells, that is when you start having these compounds now being released into the bloodstream, and it will come in such high concentration. So things can accumulate today and later in old age begin to result into some complications. Some people will begin to develop some central nervous system conditions because of the exposure to some of these substances. All right, so that's just a very practical example, how to apply some of this knowledge to real life, especially with that knowledge of how you can have some of these substances acting as irreversible inhibitors of enzymes, which really can affect the central nervous system, which will result into paralysis. And of course, I think DIFP sometime back was even used in chemical warfare. So it was used in some chemical weapons to cause paralysis in soldiers and stuff like that. And many nerve agents, that is the purpose for it, to cause paralysis. Once there is paralysis, you can't move, you can't do anything. Obviously, the nervous system, when it's affected, can result into fatality when muscles are not moving. It's not just the muscles of the limbs, it can result into paralysis of the muscles of the heart. And if the blood is not being circulated to all cells around the body, then organs will start to die. And once the organs are dying, especially the brain, the heart stops. And then the end of it is the whole organism also dies, because there is no blood flow to all the cells for proper metabolism to occur in all these cells. All right. Now, another example is to do with the activity of penicillin, common antibiotic, penicillin. So you remember we talked about the cell wall of bacteria being made up of peptidoglycan. And peptidoglycan, from first year, we saw as being a polymer of derivatives of monosaccharides, N-acetylglucosamine and N-acetyl muramic acid. Even when we started this semester, we saw that structure of N-acetylglucosamine and N-acetyl muramic acid binding together, having that polymerization to give you the cell wall of bacteria being peptidoglycan. And that was when we were talking about the enzyme lysozyme, which is responsible for breaking the glycosidic bonds between these monomers, N-acetylglucosamine and N-acetyl muramic acid, resulting into breaking the cell wall of the bacteria, and thus resulting into a bactericidal effect. Now, in this case of penicillin, we have the enzyme glycopeptide transpeptidase. So the name will tell you quickly that, okay, this is a transferase. And this transferase acts on, or is having a substrate, which is what? A glycopeptide. Or you can say maybe the product is glycopeptide, because transpeptidase. Transpeptidase means it is transferring peptide. So it's responsible for transferring some peptide onto the the monosaccharide molecule to give you that derivative of the monosaccharide. So it is in that process of polymerization of N-acetylglucosamine, N-acetylmuramic acid to give you that, compound peptidoglycan, which is the cell wall of the bacterium. That is where you have this enzyme, glycopeptide transpeptidase acting. Now, glycopeptide transpeptidase is what is depicted in this curve, like yellow. Like this is glycopeptide transpeptidase. It's an enzyme, and that enzyme is having serine residue in its active center. Remember, chymotrypsin was also having serine residue. So you can see that serine residues are really reactive because of the hydroxyl group that they have on them in the side chain. So they are really reactive. And so you find them, a lot of times in the active center of enzymes. We mentioned the amino acids, which you could find in the active center of enzymes in one of our previous lectures. You can go back and do a revision of that. So you see serine, which can bind the hydroxyl group can react with penicillin. So penicillin, you see the penicillin compound, you have a carboxylic acid group which binds covalently to that side chain of serine in the enzyme glycopeptide transpeptidase to give you a complex of the penicillin and that enzyme. That is a covalent interaction between penicillin and the enzyme. And by that, rendering the enzyme inactive. Then if the enzyme is inactive, it means the enzyme cannot act in that process of synthesis of peptidoglycan, which is the cell wall of bacteria. And then that, of course, will result into a bactericidal effect, or let's say, can say lysis of the bacterial cells, the breakdown of the bacterial cells. Now, you see, if this was I'm teaching. If this was, say, toxicology, that sort of reaction between penicillin and the enzyme, we refer to as something like a lethal synthesis, or for instance, or maybe there's another term which is used, say, suicide metabolism. So you see, such an inactivator as penicillin can be referred to as a suicide inactivator. Of course, it is inactivating the enzyme, and that inactivation is resulting into the death of the bacterial cell. And so you have that being referred to as suicide inactivators. And the reason why in let's say in toxicology, I'm saying it can be referred to as, this process can be referred to as lethal synthesis or suicide metabolism, because it's some form of metabolism. It's synthesis. And what is synthesis? Synthesis is anabolism. It's a build-up of bigger molecules from smaller ones, which is referred to as synthesis in metabolism. So you see that happening with the activity of penicillin. So, I mean, these are some specific examples of how inhibition is used, as we saw earlier in the design of pharmaceutical agents. So these pharmaceutical agents actually act as inhibitors to some enzyme which is responsible for the catalysis of an enzymatic reaction, of a metabolic reaction, and by that stopping that metabolic reaction, and thus having some control, some regulation of a specific reaction, which will then give some therapeutic effect. All right, so in summary, in general, what we have gone over today is, types of inhibition. We divided this into reversible and irreversible inhibition. And reversible inhibition, we divided it into competitive inhibition, non-competitive inhibition, mixed inhibition. And we also look at irreversible inhibition, and we look at some specific examples of irreversible inhibition in terms of pharmacotherapy, and even in terms of pest control. I mentioned how this knowledge is used in controlling pests when you have some chemical agents being used as nerve agents causing paralysis in these organisms, and thus rendering them inactive. So that brings us to the end of the lecture. I will now allow for questions or contributions. Anyone who has anything to say, you can put up your hand, and I'll call your name, and then you can say what you want to say. So the floor is now open. So if you want to say anything, you can just put up your hand, and I'll call your name, and you can say what you want to say. Yes, Ibrahim.
Sir,
Yes.
Please, do enzymes normally remain after the catalyzed reaction?
Do
Like,
enzymes normally remain after they
have
catalyzing a reaction?
Yes, sir.
Yes. The enzyme itself does not get converted together with the product into another entity. The enzyme only catalyzes the reaction. It only comes in to, as we saw earlier, decrease the activation energy of the reaction. And once the reaction is done, there is dissociation between the enzyme and the product. The product is given out, the enzyme is left free and can continue to catalyze the reaction until all the substrate that is present for the reaction to occur is consumed. So yes, the enzyme will remain after the reaction is done. Any other? So if you have anything to say, you have questions, or then you can. We are all ears. Okay, I can see maybe there is something in the chat. I always give my slides, a dark wire rough maths. I always give out my slides. So right after the lecture, I'll send the presentation to the class rep, and he will share it with you. So, and this particular lecture is being recorded. So after the lecture, I will also send the video. So if you want to watch, you can watch. So what I would need is Wisdom. You need to send me your email address, because the video is it has a bigger size than the normal, files that I can send.
All right.
I will send a video through email. So if it's on a Google Drive and you share it, anybody can watch it.
Yes, sir. All right.
So anybody wants to say anything? Question, contribution? It appears everybody is tired. But today your number is low. Wisdom, how many of you are in the class?
About 30, 37 then.
About
If
37.
some has add up, I would have to update my list, but the old
Okay.
list I have is 37.
Oh, okay. Then that's
Yeah.
all right. You are 31 people online now. So if it's 37, then it's all right. But it looks more like there are more people who are okay. Maybe it is.
Okay.
I'm confusing you with the weekend group. I think the weekend group is much more than.
The weekend people, they are many power.
Okay.
Yeah. Yeah.
I see. Musa, I can see your hand.
Good evening.
Good evening.
Yeah. Please, what is the role of the competitive inhibitor during enzyme?
What is the role of the competitive inhibitor where?
During enzyme action?
During enzyme action.
Yes, please.
Well, the competitive inhibitor, if it is present, it only comes in to regulate the rate of the reaction. If there is a competitive inhibitor, then it's just regulates the reaction, because it can easily be undone if it is there. So what happens is a lot of biochemical reactions in the body have these different mechanisms of regulation, and many of them are by what we refer to as feedback inhibition.
Okay.
And by that, what it means is that the final product of the biochemical reaction actually acts as an inhibitor to an enzyme along the reaction path. So, for instance, if it is the process of synthesis of hemoglobin, either in the liver or in the erythroid cells of the bone marrow, you will see that the hemoglobin molecule itself will act as an inhibitor to an enzyme which is acting along the reaction path of that process of synthesis. So the feedback mechanism is like there is a product which is being produced in a whole chemical, reaction path. And once there is enough of it, when you have high concentrations of that product there, then it's giving signal back to the enzymes that we are enough. There is no need to produce more. So that inhibition is some sort of reversible inhibition, because it can be undone. Once you have the concentration going lower, then the reaction will start again. So you can have that form of inhibition occurring in many biochemical reactions. So that's just one example of how you see. So like synthesis of hemoglobin, even in the activity of hormones, for instance, they also have such feedback inhibition. When there is some effect, there is some production, some of the hormones only cause the release of another hormone. And then they're supposed to have some specific effects in regulating some particular chemical reaction. Once the effect is achieved, then you see that you have the product now acting as an inhibitor. And then once the concentration goes down, you have the release of that inhibitor away. You find out not only in vertebrates, not only in human beings, in bacteria, you see in microorganisms, you see similar activity, similar ways of regulation of chemical reactions, even in microorganisms. Yes. Any other? Musa, is it clear?
Yes, please.
All right.
And I would also like to know the significance of competitive inhibition to
The significance of
competitive inhibition in enzyme.
I didn't
enzyme
hear you well. The significance of what?
Competitive inhibition
Mm-hmm.
in the enzymes enzymology.
In enzyme what?
Hello, can you hear me?
I can hear you. I didn't hear the last part. The
I
significance
said that
competitive inhibition in enzyme what?
enzymology.
In enzymology.
Yeah.
Oh, okay. So what I mentioned right now was, I think you that's the same question you were asking.
The same question. Okay. Okay. All right. Thank you. Thank you then.
All right.
Okay.
Any more? Well, it appears nobody wants to say anything. Musa, your hand is still up. Or it's just that you've forgotten to put it down. All right. So if there is no question, no contributions, then I will move the motion for our meeting to be adjourned till we meet next week, Monday, same time. So all those in favor, people are even dropping out. All right, then.
How do you
The
do?
best.
I second.
See you next week.
All right.
For now.